Good evening, everyone. Welcome to the next lecture in the distinguished lecture series at Ashoka University in the Trivedi School of Biosciences. We're very pleased to have Dr. Jitu Mayer today. Jitu, as you may know, is currently the director of the National Center for Biological Sciences in Bangalore. He also is a member of the advisory panel for the Trivedi School of Biosciences and has been advising us from the very beginning. Of course, many of you know Jitu as a leading biologist, not just in India, but internationally. And I'm not going to go into all the prizes he has won and all the distinctions that he has received. But just to say that if Jitu had paid enough attention to his cricket than he did to his studies, he could have been playing for India. Beyond that, Jitu and I have worked together. We have collaborated and we've had many discussions on various things, science and otherwise. And I can honestly tell you that he is one of the most likable people and one of the most knowledgeable people. So I think, Jitu, that's enough embarrassment for you. And I'd really like to invite you to give this talk. So it's over to you, Jitu.
Yeah, can you hear me, Shahid?
Yes, yes.
Yeah, and see my slides as well?
Yes.
Okay, great. So thank you, Shahid, for that. It was, it's actually great pleasure to be encountering you again. As you know, we have worked together, and it's been a great pleasure for me to give this talk at your behest. So what I'm trying to say is that blame is yours. And the talk today is really about how physics in a very fundamental way has been a motivator for much of what we understand about biology today. And of course, it doesn't need me to belabor the point of how the molecular biology revolution itself was cast by physicists who were intrigued by the molecules of life. But I think we are also in a new revolution of the engagement of this encounter of physics meeting biology. And I'm going to tell you how I'm seeing that revolution of the engagement or this encounter of physics meeting biology in the areas that I've been working on, which is the cell membrane. So, so this membrane of the cell, and I'm going to give you something like an understanding of where we stand with our current knowledge of the structure and organization of the membrane of the cell. The cell I'm going to be talking about in the main is the eukaryotic cell membrane, but I'm going to refer to other membranes as well. Now, the eukaryotic cell membrane, as most of you know, is this really, in this view graph, it's outlined here. It's you're looking at the surface of a lowly Chinese hamster ovary cell in a tissue culture dish. But if you look a little closer at its membrane, it is ensheathed by a 4 nanometer layer, which covers this entire surface, consisting of a huge number of different distinct components, proteins and lipids, if you will. Now, the understanding that the membrane of a living cell was this colorful or this picturesque was actually not how it all began, at least how our understanding of the cell membrane began. And let me first, bring you up to speed with a little bit of history of how we begin to understand the structure and organization of the cell membrane. But I must say, this was the first encounter of, of physicists actually trying to explore what biology was about and how could biological systems exist in a way. And the membrane, I think, of course, since it's at the outside of the cell, posited itself and presented itself very kindly to the gaze of physicists. And the journey began, if you will, when Agnes Pockels built a Pockels cell, which eventually led to the Langmuir-Blodgett film, trough, where then Gorter and Grendel took red blood cells, which are cells that are enucleated, extracted lipids, and threw them onto this Langmuir-Blodgett trough, and discovered that the area that the lipids occupied were, in fact, because in this Blodgett film, you'll make a monolayer, and the area that the lipids or the monolayer occupied was, in fact, that of twice the area of the blood cells that they had extracted the lipids from. Well, this, of course, led to the positioning or positing of the idea that the bilayer, that the membrane was, in fact, outer membrane of the cell was, in fact, a bilayer. And by then, bilayers of various amphiphiles had already started being noticed in the physics community. I should say physics and chemistry, in some sense, are very much aligned at this stage. The idea that there were proteins in the bilayer were then, brought about by Danieli and Davison, where they proposed that, this fantastic model that proteins were sort of adsorbed onto the two surfaces of the bilayer. But then, it took another 40 years, if you will, with Singer and Nicholson looking very carefully at the distribution of membrane proteins in electron micrographs and studying and understanding data from Michael Edidin's work that proteins and lipid diffusion, could be combined with sound thermodynamic principles to give rise to what is today the Singer-Nicholson model shown at the bottom left. The Singer-Nicholson model simply states that you have lipids, that proteins are dissolved in a sea of lipids, and things are well mixed and moving about in happy equilibrium. So given this, we needed then to confront this other beast, which is the fact that the membrane of a cell, which was already begun to be characterized to death by respectable chemists, that phospholipids and glycolipids formed the main constituents of the membrane. And there were, maybe over 1,000 different protein and lipid species in the membrane. And in a eukaryotic membrane in particular, one found this creature, the cholesterol molecule. Okay, so now you have this drab picture of Singer-Nicholson on the right, and you have this sort of array of different compositions on the left. How is all this put together? How is it organized? And this became sort of, in some ways, the preoccupation of our physicists and chemists friends for once this synthesis of Singer-Nicholson came about. But the question we needed to ask, of course, is where do these lipids come from? And here is where biology encounters everything else. And it says that nothing in biology, and this is Dobzhansky speaking, nothing in biology makes sense except in the light of evolution. And of course, evolution brings us to the three kingdoms of life: bacteria, archaea, and eukarya, which are being recognized by the 1970s. And I must say that these three kingdoms of life give a understanding. I'm just sorry. My dog is demanding something. I'm going to take a little break here. Just a second. Yeah, sorry about that. I think he woke up and needed some food, like all of us do. It turns out that the ancestor of all cells the common ancestor of all cells, must have had a very complex system. It gave rise to bacteria, archaea, and eukarya. And the idea that eukaryotes came about from a mixture of bacteria and archaea is something that is already a well-established feature. And we know that it's well-established because the DNA replication machinery that all eukarya, that is all of us here tucked away in this little corner here, share, the DNA replication machinery is obtained from our DNA replication machinery, or eukaryotic DNA replication machinery, obtained that from the archaea. But what about the lipids? And if you look at the lipids, it turns out that the lipid species that we carry are similar to those that are present in bacteria, not in archaea. And this, in fact, was quite a puzzle, and it became an even bigger puzzle because when you look at the synthetic machinery that's present for all lipids in bacteria and eukarya, as opposed to archaea, one realizes that the archaeal lipids, in fact, have the opposite chirality of their glyceraldehyde or their glycerol backbones that make phospholipids. Over here, they have a chirality that is actually literally opposite that of the chirality of the lipids that are present in bacteria and eukarya. So we've got lipids from bacteria, which have the characteristics of the bacteria, and we have the chirality of which has the appropriate chirality, and we have the DNA replication machinery from archaea. So this is itself a puzzle, and it remains an unsolved question today of how did the eukarya acquire these lipid synthesis potential that resembles the bacteria but not the archaea. So perhaps there are archaea lurking there that have bacterial-like lipids, but that seems to be a vanishing possibility so far. What is even more interesting is that archaea lack sterols, and sterols are an essential part of eukaryotic membranes, and some bacteria also carry sterols, at least some aspects of sterols. Sterols emerge, and in fact is a defining feature of eukaryotic emergence. Sterols emerge around 2.7 to 2.4 billion years ago, pretty much, after about 1 billion years after, over 500 million years after the emergence of the early forms of life on this planet. And they emerge because of a climate change. They emerge because the oxygen is being now enriched in the atmosphere by oxygenic photosynthesis, and the oxygenation of the atmosphere and oceans is contributing to the development of an oxygen tension, an oxygen pressure, if you will, in the atmosphere. And this is essential to make cholesterol. So until now, bacteria lived without cholesterol. But then once oxygen began to be available, the synthesis machinery allowed the cyclization of the structure that cholesterol today exhibits, which is a polycyclic group shown right here in the middle of your screen, which is present in all vertebrates and in enormous diversity in all eukaryotes. And there's a fabulous statement here from Konrad Bloch, who actually first synthesized the cholesterol. He says, selection pressures rendered the sterol molecule progressively more and more and optimally competent for membrane function. In fact, the sterol pathway terminates with cholesterol, a molecule designed to optimize attractive van der Waals interactions with phospholipid chains in the membrane. And this, I would argue, this principle, I would argue, gave rise, if you will, to the second encounter of physics to what supposedly was a solved problem, that is the structure of the membrane in the Singer-Nicholson framework. And here, this was the sort of introduction of the idea of physico-chemical expectation of phase segregation. So if you take lipids in a bilayer, they can exhibit multiple states depending on the temperature at which they are. And if you throw cholesterol into that mix, cholesterol helps to freeze the acyl chains of these lipids and create sort of better interactions between saturated acyl chains amongst themselves to create a liquid ordered region in the membrane. And otherwise, these saturated acyl chains at reasonable temperature are in fact in the disordered state. And the gel phase is thought not to exist in to support at least in equilibrium with the liquid crystalline phase, because it may cause defects and create leakages. So if you want to sort of, if you will, a semi-permeable system, the options one has are disordered membranes in conjunction with liquid ordered membranes exhibiting some kind of a partitioning between the two. And this requirement was motivated again by biology, because in the early 80s, it was realized that polarized cells, for example, exhibit very distinct lipid compositions at the apical surface of the polarized cell versus the basolateral surface. They, in fact, had distinct lipids that were present in one or the other surface. And the idea began to emerge from the fact that membranes could phase segregate with the assistance of cholesterol in these liquid ordered and liquid disordered phases. The idea that membranes could segregate gave rise to the view that you had membrane domains or rafts, which were sort of functional heterogeneities in the lateral distribution of these complex mixtures that began to look a bit like this patchwork quilt shown in the bottom left, from where molecules could accumulate and then be distributed to different domains in the cell membrane or in the cell at the cell surface through sort of picking and plucking different regions of the membrane to deliver them to different parts of the cell. And what we do know, and this is now some really wonderful work over many, many years, that the composition of the cell membrane, of the membrane of cells, especially in a eukaryotic complicated cell like a eukaryotic cell, the endoplasmic reticulum has a distinct composition, the mitochondria has a distinct composition, so does the Golgi, and of course the cell surface has an even more distinct composition. And there's a gradient of cholesterol that goes from the inner leaflet to the outer leaflet, suggesting that, of course, that there must be some sort of a gradient of segregation that also takes place dependent on cholesterol in these membranes. And in fact, proposals that there are distinct phases in the cell membrane began to abound to explain much of our experience with the biology that was driving the expectation that one could see sorting and segregation of lipids contributing to the functional sort of segregation of molecules in a living cell. So, so of course, given this, the injection of this idea of lateral heterogeneity into this humdrum Singer-Nicholson model, one had to ask the question: is there evidence for this? And of course, the only way to ask the question about evidence for this was to ask the living cell whether the membrane of the living cell was partitioned into different phases or not. Or could one see evidence for such partitioning? And this is sort of a work that I had begun now more than 30 years ago when I was a postdoc in a laboratory in New York, looking for the organization of these molecules called GPI anchored proteins, which was supposed to partition into these liquid ordered domains. So to explore their organization, I put little, binoculars on these GPI anchored proteins to tell us something about the organization of these GPI anchored proteins at the surface of living cells, and asked the question whether we could indeed observe, if we observed these GPI anchored proteins, they would tell us about the organization of these raft phases in the membrane of living cells. And so once we put these GPI anchored proteins onto the surface of a living cell, what we found was, for example, here is a fluorescently labeled cell which carries a GPI anchored molecule. And by looking at the distribution of the GPI anchored molecule, and what we found was that it was remarkably uninteresting. It was flat, completely boring in its intensity. Now, just as I was about to leave this field and move on, I said maybe there could be something that the GPI anchored proteins tell us besides the fact that they're uniformly boring at the cell surface. And this was necessitated by the observation that GPI anchored proteins actually are sorted both in the inside the cell as well as the cell surface, and they get into distinct endocytic carriers or endocytic vesicles at the cell surface. And so there must be something more going on. And using a method that we developed when I had moved to Bangalore to set up my laboratory, we found that the GPI anchored proteins distribution, although at the long length scale, at the scale of the optical resolution, was extremely uninteresting, at the resolution of nanometric interactions, it was extremely interesting. So the GPI anchored proteins formed extremely tiny clustered patches in the membrane, revealed by a method that we devised called FRET imaging and FRET microscopy, where we can visualize the fact that the GPI anchored proteins form tiny little nanoclusters in the membrane of living cells, concentrated in these patches and heterogeneously distributed to the cell surface. So finding this was almost like finding, in a desert, suddenly a castle which was sort of made out of these extremely ephemeral dust particles, and almost an impossibility. So this, of course, intrigued us. And when I say us, it was Madan Rao, a colleague who'll show up later in this narrative, a little more detail. And what we found was that the distribution of the GPI anchored proteins was extremely unusual. It was spatially heterogeneous, as I've just shown you. It's formed these clusters independent of their concentration. The clusters themselves happen to be immobile, and they were sensitive to the concentration of cholesterol in the membrane, something that the rafts also exhibit, concentration of cholesterol. But these clusters were rapidly remodeled. What was even more intriguing was that these GPI anchored protein clusters were actively maintained by the interaction of membranes of eukaryotic cells with their cortical cytoskeleton. So the membranes of eukaryotic cells, if you remove them from their cortical cytoskeleton, as Darius has done here in this movie, where you make cell blebs, which are removed of the cortical cytoskeleton, the clusters of the GPI anchored proteins simply disappear on these blebs. Now, what do we know about the organization of the actin cytoskeleton under the membrane? And at this time, my colleague in Japan, Aki Kusumi, had produced some beautiful images of how the lipid anchor, how the bilayer of the cell was sort of stuck onto this fenestrated cortical actin meshwork. And this cortical actin meshwork actually impacted the distribution and diffusion of molecules in the membrane as they encountered this so-called static mesh that provided a mechanical scaffold to the GPI, to the outer membrane of the cell. Now, this mechanical scaffold was something that was passive, did not really do very much, and we soon realized could not provide a motivation to build these clusters of the GPI anchored proteins, because the GP anchor proteins required an active element of the cytoskeleton. Some activity was required. So this is where I would argue the third encounter with physics happened. And now I think this is perhaps a very important encounter, because the physics of active systems that we are beginning to see is proliferating in biology today. And it's providing a really extremely powerful explanation of many things that we encounter in biology. And I think for us at the membrane has been an incredibly valuable encounter. So for at least us who sit on this thin, flimsy 4 nanometer fabric of the cell, it's a very important encounter. I'm going to tell you why and how. So, and of course, this encounter was enabled by collaboration with my dear friend, colleague Madan Rao, who is a soft matter physicist. In fact, he's a strong exponent of this active matter business, and is somebody who really has been pushing this field for a while now. So what he and his student Kripa proposed was that perhaps the clusters are being built by their engagement at the cytoplasmic leaflet of the cell by a sort of a meshwork of actin filaments and motor proteins that are present in this, invisibly, if you will, in the underbelly of the cell membrane. Of course, this because they were physicists, they could make bold statements like this. Any sensible biologist would have been laughed out of the room. But being physicists, they did the next thing, which is they set up a theoretical framework to tell us what would happen if such actin filaments were present at the underside of the cell membrane. So they said, let's consider 2 species of actin, the short filaments that they propose, and of course the static mesh that we and Aki and all can see so easily, and imbue both of these systems with motor activity. Motor activity would give the short filaments multiple possibilities: treadmilling, alignment, advection, and of course contractile stress coming from the static mesh, as well as these filaments. This configuration would give rise to these 2 dynamic aster-like configurations in the membrane. I'm going to now just point out a set of equations that they wrote down. These equations, reflect the equations that describe the orientation of the filaments, which contain terms that come from energy-consuming mechanisms, that is relative sliding due to the myosin motors, contractility of the filaments, again, dependent on the concentration of the filaments and the interaction with the motors. They also describe the filament concentration, which is, of course, a consequence of the concentration of the filaments itself, but that concentration now is a consequence of the active advection governed by the consumption of energy because of the treadmilling of the filaments and their engagement with these motor molecules. Of course, the elements that would always exist would be things that should, in fact, be present in the membrane and present in even in a dead system. And these systems now, these equations, which also, by the way, describe the flocking of birds or the formation of these beautiful patterns of fish in the ocean, the same equations also describe the flocking, if you will, of these contractile filaments. They added several terms that were specific to our system, which was sort of a term that contributed to the active noise, which was basically looking at binding, unbinding, the stochasticity of binding, unbinding at these molecular scales, and the stochasticity of length distributions of the filaments. And what they found were these patterns that, to my undisguised, were sort of, critical eye, look very much like the patterns that we were seeing of our GPI anchored proteins at the cell surface. So it was this very simple fluctuations of these actin filaments that gave rise to these patterns. Okay, so if this were true, of course, the model recapitulated the key features of the cell experiments. They explained in experiment on left, theory on right, the heterogeneous distribution of the clusters, the non-random variation of cluster size distribution, all the physical parameters that we could actually measure of these clusters. They exhibited sort of and could determine that you could obtain a very specific non-Arrhenius remodeling kinetic of the clusters themselves, and a concentration independent clustering of the molecules of the clusters themselves. But not only did it explain the observations that we had made, it also made predictions. First of all, it said very simply that anything that is coupled to the actin, this dynamic actin at the underbelly of the cell membrane, should indeed form these clusters. So if you had transmembrane proteins, not GPI anchored proteins, but transmembrane proteins, which had actin binding capacity at their cytoplasmic leaflets, this clustering machinery should indeed produce clusters of the transmembrane proteins. And in fact, it did produce the clusters very much like the clusters of our GPI anchored proteins. It should also drive active signatures of the fluctuations of the intensity distributions of these molecules. And for the physicists in the audience, it was clear that if you look at the fluctuations of molecules you can predict diffusion, you can predict many things, you can predict also the nature of the underlying medium. And by looking at the fluctuations of the intensity of the distribution of the molecules, the theory predicted that we should see fluctuations that are very far away from what you'd expect in a membrane that's being simply buffeted by thermal influences. So at thermal equilibrium, the fluctuations in the membrane would scale as root of the number of molecules. I mean, this is standard physics, but with activity, they predicted that the fluctuations should scale at a very particular exponent. And in fact, in experiment, that's exactly what we saw. And with the activity, you had the scaling, which went as the 0.8 to the power of n, whereas without the activity, it looked exactly like a membrane at equilibrium. So we said, well, okay, the theory seems to be very powerful. It makes predictions. But if it's really that simple, if it has just 3 elements, myosin motors, filaments, and the filaments are now interacting with something in the membrane, we should be able to recreate it. And in fact, harking back to Feynman, what I cannot create, I do not understand. So we said, well, if we really understand this, we should be able to recreate this. And this time, 3 intrepid colleagues of mine, Darius Costa, Abrar, and Kabir, they came and said, well, Darius actually showed up first and built the membrane bilayer on which we could attach a linker that could associate with actin filaments. And we put in actin filaments that were freshly polymerized, and through sort of bi-headed molecular motors from skeletal muscle onto the system. And lo and behold, what we saw was this remarkable view graph that Darius generated of actin being clustered by myosin in the middle and clustering the lipids that it were associated with on the right. And this now, if you throw in the length distributions of the actin and a continuous source of ATP, gave rise to this extremely dynamic remodeling system that we think recapitulated the membrane that we were seeing. In fact, the physical measurements that we made on this in vitro system very closely matched the measurements that we got on the cell surface. So armed with that, we said, well, all this looks very plausible, and let's go ahead. But then there was a small problem here. We discovered that GPI anchored proteins are not like transmembrane proteins that have a direct link to the actin, that can be endowed with a direct link to the actin. In fact, GPI anchored proteins sit on the outer leaflet of the cell membrane, whereas the actin sits on the inner leaflet. And the question was, how do these things couple across the bilayer? How does the actin at the inner leaflet connect to the GPI at the outer leaflet? And here we teamed up with a really long-term chemist collaborator of mine, Ram Vishwakarma, who was at that time at the IIIM in Jammu. And he and his group of chemists, including Parvinder Singh, along with two students of mine, Ambika and Ria, they synthesized these very difficult molecules, these GPI anchored proteins with different lipid tails and fluorescent tags, and send them across to us from Jammu for us to explore whether these molecules could be clustered in the membrane. And what we found was that the only thing we needed for a fluorescently tagged molecule to be clustered from the outer leaflet was if it had long saturated acyl chains, if it was endowed with long saturated acyl chains, and it had cholesterol in the bilayer. So, so now, given that we had this consideration of the long saturated acyl chains in the outer leaflet, what could be the coupling lipid at the inner leaflet? And this is where we discovered that phosphatidylserine was an endogenous lipid that in fact has long saturated acyl chains naturally occurring at the inner leaflet. And by the way, I should point out that the GPI anchored proteins also have naturally occurring long saturated acyl chains at the outer leaflet. We'll get to that in a little bit. So now we had phosphatidylserine at the inner leaflet with long saturated acyl chains available. And if we took cells that were unable to make phosphatidylserine and then reconstitute, they of course lost their clusters of GPI anchored proteins. But if we now added back these lipids with saturated acyl chains, they recovered the clusters of the GPI anchored proteins. Now, that told us that of course you needed saturated lipids at both ends, and if they were in some sense encountering this actin at the inner leaflet, they would build clusters. So to explore this in a little more detail and more quantitative terms, of course, Madan came to the rescue, and he and Anirban set up atomistic simulations of asymmetric membrane bilayers that had phosphatidylcholine, the sphingomyelin, and cholesterol at the outer leaflet, peppered with some GPI anchored proteins that had different acyl chain lengths. And of course, when the cell is at temperatures above the phase transition temperature of these lipids, the membranes themselves are extremely well mixed. In fact, as you will see in this depiction here, this is a bilayer, asymmetric bilayer at relatively high temperatures. The membrane is well mixed. And indeed, this is the bilayer now at the down here at the lower part of this view graph. You see what we have done here is just held the PS lipids and held them in a position. And when they are held at this high temperature, they now attract the GPIs in red at the outer leaflet and construct these clusters. And this immediately told us that what was happening was that the GPIs were perhaps being held across the bilayer by their constraints with the interaction with the actin cytoskeleton at the inner leaflet. And looking carefully at the lipid structure locally, we were building liquid ordered patches in this membrane because cholesterol was involved in creating these liquid ordered patches here. The lipid chains themselves exhibited ordered characteristic in the atomistic simulations. And so that we felt was a handle for us to now ask in experiment, what would we see? So the prediction was that if we had this operation going on of molecules being held at the inner leaflet by these dynamic actin cytoskeletal machineries, they would form these clusters, and these clusters themselves would exhibit liquid ordered patches. So that in some ways was another experiment that was suggested by some of the simulations. And in order to test that, we set out to ask ourselves whether we could actually detect these ordered domains in these membranes where these GPI patches were present. So we turned back to chemistry again, and looked at probes that could distinguish solvent permeability in membranes. These probes are probes such as that of a molecule called laudan, which, if present in a liquid disordered patch, exhibits a fluorescence that is red-shifted. And when it is present in an ordered patch, where solvent penetration is reduced, in fact, that's a signature of an ordered patch, the fluorescence emission of this laudan is somewhat blue-shifted. So Trivedi in the lab then decided to explore this correlation, and he found that where the clusters of the GPI anchored proteins were present in the membrane, the membrane tended to be exhibiting more of an ordered exposition. So we believe for the first time, we were beginning to see these regions in the membrane that were ordered, and they were ordered now because of activity. And I think this is a very important process. They're not ordered because the molecules were phase segregating due to their propensity to phase segregate due to thermodynamic principles, but they were segregating because they were now being held and driven by this activity and forming these ordered patches. So this led us to the idea that the heterogeneity of membrane organization was now a consequence of this dynamic nanoclustering templated by the dynamic actin cytoskeleton at the underside of the cell membrane. And any protein that had an actin binding domain would be subject to this, and lipid anchored molecules that could interact with PS at the inner leaflet would also be subject to this process of clustering, giving rise to these mesoscopic domains. And the mesoscopic domain itself is a consequence of this activity that generates this nanoscopic feature. So the membrane then can be now thought of as a membrane actin cortex composite. The molecules that don't interact with this composite, we believe, will obey what Singer-Nicholson had postulated of a well-mixed system. The molecules that interact with this dynamic actin cortex are going to generate the structures at the nanoscale and at the mesoscale I just talked to you about. But now this gives rise to a very important feature, that this entire process of organization can be regulated. So we believe that this mechanism is going of being able to deploy activity or active principles are going to contribute to regulation of the organization and structure of the membrane. And I'm going to spend the next few minutes towards the end of the talk telling you about how this happens. So, so what we realized was that many membrane proteins, and we now focus on one, the Integrand receptor, which senses the outside world. It senses the chemical nature of the outside world through its capacity to bind the extracellular matrix. It also activates signaling in the inside of the cell and exhibits a capacity to modulate the actin cytoskeleton. And it turns out that the very molecules that it uses to modify the actin cytoskeleton are involved in generating the actin and the myosin that is necessary to build the clusters of these GPI anchored molecules. So if one actually deletes these GTPases and the signaling system downstream of the integrin molecule, you actually get rid of this entire clustering phenomena. What we also realized was that the integrins required a mechanical transducer to also support the formation of these clusters. Now, one may say, well, this is all very well. You've discovered how the membrane is building these membrane rafts, but for what use? I mean, why would it matter for the cell? And I'm going to provide you now some physiological context for this. So if you look at this, one of these cells that express our GPI anchored protein and watch that cell now being dropped down on a surface that carries ligands for integrins, what you see is a remarkable thing, that the cell itself forms, is arriving at the surface of the ligand coated surface without any clusters. But moment it starts beginning to expand, it seems to generate these clusters in situ as it arrives on the cell, on the surface. And you can now track that in real time, because these measurements that we do with these GPI anchored proteins can be tracked in real time, and you can track it quantitatively as well. And what we find is that when the cell arrives at the surface of any extracellular matrix component, it seems to build these clusters as a consequence of the engagement with the extracellular matrix. And these clusters then, we believe, are contributing to the capacity of the cells to spread. So to ask whether these clusters are indeed doing so, we took advantage again of some genetics, where one could mutate the enzymes involved in building these long saturated acyl chains of the GPI anchored proteins that I've been telling you about, and asked whether upon mutating the capacity of the cells to build these long saturated acyl chains and create these clusters, because now the GPI proteins will not interact with the inner leaflet, will they in fact exhibit such sort of spreading reactions? And in fact, what we find is that the clusters of the GPI anchored proteins shown here as these warm colors are completely disrupted in the mutant that is unable to build the clusters unable to in fact extend the saturated acyl chains of the GPI anchored proteins. And in fact, the membranes themselves exhibit very, very poor ordered domains as well. And in fact, what happens is that the cells are themselves unable to spread. Not only are they unable to spread, they're actually unable to crawl or move. And so the clusters, we believe, are fundamentally important in doing so. And I think this comes to the possibility that signaling is now building clusters through the deployment of signaling reactions at the inside of the cell. For example, the activity of the GTPase0A, which then contributes to the construction of these liquid ordered patches. These liquid ordered patches then instruct the membrane to do other things. For example, they could recruit molecules that are involved in activating the spreading reaction like RAC, and then the cell starts spreading. And of course, these are things that one would like to look at in the future, but effectively, what I've shown you is that signaling receptors using the machinery that I've just described that are available to them to control the actin cortex are able to sculpt the local membrane environment for their functional purposes. So we are seeing quite a fundamental shift from the membrane as a sort of a passive, well-mixed system, that of the Singer-Nicholson model, where the membrane itself is simply holding proteins, allowing proteins to do their job, to the membrane going from being one of structured by the thermodynamic principles of the interactions of the molecules themselves, exhibiting phase segregation due to the propensities for their segregation, to a phenomena which I think is more relevant to an energy consuming system like a cell, which is that the actin cytoskeleton, which is at the inner leaflet of the membrane, dynamically and intimately interacting with the membrane lipids, constructing a variety of different structures. What I've talked to you about today is, of course, one of these structures, that of the little nanoclusters, which can then pattern the membrane to form much more chemically significant structures like these ordered patches, which I believe are what the membrane grafts are reflective of. The lateral heterogeneities that were thought to be determined by the physico-chemical properties of membrane composition are in fact generated by their interaction with the activity in the cell. Now, this has many implications, that it allows the cell to locally control composition. It provides regulated assemblies. I think it can do active chemistry, which is a whole area that one can explore. And of course, it's going to be most important in information processing, because now we've injected energy into the equation and organization and structure. So I think in conclusion, when physics meets biology of the cell surface and through these multiple encounters, I think what we have seen reflected is an intertwined relationship between concepts borrowed from physico-chemical studies on artificial membranes, fundamental insights from theory, as I have just described to you, and of course, with reality checks, with experiments on membranes of living cells. And I think that's where we need to be focusing on what's happening on the membranes of living cells and whether we can understand them from our encounter with the physics and chemistry of today. And one cannot ignore evolution. It had so many open questions there about how these compositions arise, what are their roles, what are their functions. And I think this is sort of the exciting part of the future today. So I should just end by saying the cell surface is not this flimsy, well-mixed lipid milieu, but in fact, it's a living, breathing skin that drapes the skull, consuming energy, creating structure and organization that instructs many aspects of what happens at the cell surface. And in fact, it gives rise to the following paradigm that the membrane itself is orchestrated by these active elements, and that orchestration has consequences for a variety of phenomena and physiology of the cell migration, cell signaling, and I would argue even cell fate. And these all remain to be discovered. So with that, let me end by thanking the active actin membrane composite team, who's currently or just currently in place. Here's Madan and his colleagues Kripa, Amit, and Anirban, who have contributed so significantly to theory. There's Kabir, who's straddled both worlds of experiment and theory. There are our chemists, colleagues, Ram Vishwakarma and his partner in crime or in chemistry, Parvinder, and their numerous colleagues who worked with them. And of course, all the biologists who I have worked with in my lab, from Kabir to Ria, to Ambika, Suvrajit, Joseph, Chandrama, Thomas, who's somewhere here, Abrar and Darius and Bhagyashree are currently the team, as are Janvi, Parijat, Sankarshan, Saryu, and Anshul, all working at various aspects of the cell surface right now. Of course, another part of the lab studies endocytosis, and I haven't talked about their work, but I think what we are very excited by is the fact that now I think the introduction of this active chemistry and physics has really opened up the world of the membrane for us to explore. None of this work would have been possible without significant funding, of course, from the India Alliance, from the DST, from HFSP, and this very special, I want to thank this very special place, the laboratories at Woods Hole, where much of the reconstitution that Darius had begun and I had begun was actually initiated in the labs at the MBL, and many, many others who have contributed to the work. So I'm going to stop here and thank you for your attention.
Thank you very much, Jitu. That was a wonderful talk, which also emphasizes the ethos at Ashoka, which is interdisciplinarity. Biology doesn't care whether it's chemistry or physics. It actually includes everything. Those are artificial disciplines that we have created. Before I open the talk for questions, there's an announcement about the next talk. The next talk in this series will be on the 22nd of November, and the speaker would be Ron Vale, who many of you know, who was a professor at UCSF and who is now the vice president of Howard Hughes Medical Institute and the head of the Genelia Farms laboratories. And then December, we will not have any talk because most of you would be, on vacation towards the end of December. But we hope to come back in the new year with renewed vigor in these lecture series. If people have questions, they could raise their hand, unmute themselves and ask questions. Anup, please go ahead.
Hi, Jitu. Let me just turn my camera. Hi. Really fantastic talk. I've always admired your work for a long, long time. I was just wondering about this, the formation of microdomains when you drop the cell on a synthetic surface. But what if you drop it on a lipid bilayer, a supported lipid bilayer? Would it show similar kinetics and formation dynamics?
Very interesting point you picked up on. As I mentioned, if you drop the cells on a supported lipid bilayer, where the ligands are, let's say the RGD ligand that we have for the integrin is attached. I mean, you need some attachments, right? I mean, if it's just dropped on a bilayer alone, the cell simply remains a round ball and just starts blebbing. It can't do very much. But in order for it to engage, if it engages with the ligand on a supported lipid bilayer, the cell again engages with the ligand and starts to spread and then retracts. But if you provide some impediments to the mobility of the ligand, like some ridges or something in the bilayer, then you can start seeing a spreading reaction take place. And so that couples to the fact that one requires not only the engagement of the ligand and the downstream signaling that it activates, but it also requires some force-dependent unraveling of a molecule that's gatekeeping the production of these domains. And we can, of course, completely bypass that by providing that molecule to the cell. The molecule happens to be vinculin. We can provide that molecule to the cell in its opened up form where it requires no force. And now you see, a pretty robust spreading response and a pretty robust production of these nanodomains in the membrane. Yeah, so the bilayer, but if you provide increasing amounts of impediments to movement, increasing the viscosity, for example, we might be able to come to a threshold where you start seeing spreading again.
Okay, thank you.
Kasturi, please go ahead.
Hello. Thanks for the great talk. So I had a quick question about what would you expect the membrane organization to be like, the membrane energetics to be like when let's say a membrane protein is being inserted into the membrane, like things are moving from the Golgi and they're inserting receptors, let's say they're being inserted. And would you expect the membrane dynamics or energetics to be any different when, let's say, a single pass protein is being inserted versus a multi-pass protein like 7 transmembrane domain containing receptor versus like a TGF-beta receptor?
So are you talking about membrane insertion in the endoplasmic reticulum or
in the plasma membrane, like the vesicles are being trafficked containing
right. So you mean when a vesicle fuses, you mean?
Exactly.
Yeah, I know, absolutely. So at sites where vesicles fuse, they are also going to be sites where there is a lack of sort of mixing, if you will, or instantaneous mixing. So the lipid environment that the vesicle will encounter, or the membrane will encounter, will be that of the lipid vesicle, right? So it'll be a kinetically evolving membrane environment. Now, I don't think it will matter whether you have a single pass membrane or a multi-pass membrane. It will be determined most likely by the membrane lipids of the vesicle itself. So that little patch, of membrane will be determined by the vesicle lipids, and then that concentration will diffuse out. Right?
All right.
That's what one would expect. Of course, now that needs to be shown and proven. Yeah.
So, do curvature sensing proteins?
Yeah, very interesting. Yeah. So, of course, curvature sensing proteins will contribute to membrane heterogeneity. But they will all contribute to membrane heterogeneity in some sort of an equilibrium mechanism, not with characteristics such as what I described. So those sorts of things can also be seen if you sort of create bumps in the membrane and look at curvature sensing proteins and their accumulation.
The membrane with some kind of a probe or something, you can study these.
That's right. That's right. In fact, some colleagues are beginning to do that by indenting the membrane and seeing what accumulates in these patches because of the indentation. Right?
All right. Thank you.
Samelia Mishra, go ahead.
Thank you, sir. Sir, for giving me this opportunity. Sir, really, it was a very nice talk. So, I have a basic question that it is similar to the vesicle question that whenever a virus will infect a cell, when it is about to insert, will the membrane dynamics would be same related to the vesicle one, or it will be different from the vesicle insertion?
Again, a very interesting question. I mean, here, what I should mention is that just as the phenomena of the internal, stresses exhibited by the cortical actin on the membrane create structure, you could imagine that on the outside of the cell, or even on the inside, if there are molecules that will bind and aggregate components in the membrane, they will also create their own patterns and structures, right? So the extracellular matrix, the glycocalyx, etc., will pattern the membrane based on what it is interacting with in the bilayer. Similarly, a virus, which is a multivalent entity, will arrive at the cell surface and begin to pattern the very surface that is interacting with. And when it fuses, it will create the disjunction that any vesicle fusion would create, as we just discussed, in terms of a secretory pathway. So I should say that we've been looking at what happens when viruses arrive at the cell surface, what sorts of structures it can pattern, and what kind of pathways that the viruses can actually take to get into the cell.
So further, I would ask that do get any biological response, means immune response from the biomembrane system whenever a virus will infect? Any physical phenomena that will help to get rid of the virus through the biomembranes? Do we will get?
Well, I think you were probably talking about whether one could modify the membrane and see what happens, sir, in the opposite of the modification that the virus may induce.
Yes, sir.
Yeah. So people have been looking at this. In fact, for our favorite virus of the day, corona, it turns out that the virus, when it associates with the receptor, requires specific lipids to be in the vicinity of the receptor to begin to engineer its devious cascade. And when you affect those lipids, for example, cholesterol is one, you also influence the virus, the ability of the virus to infect the cell. So yes, I would say that the membrane itself can serve as a way of tuning what happens to molecules that can be taken in. Yeah.
Thank you, sir.
I don't see any other hands. So, Jitu, I had a very basic question.
Sure.
You said that intracellular membranes versus plasma membranes, the composition of lipids, or rather the ratio, is very different. So my question is, two questions. One is, what determines this? And the second is, would, let's say, a liver cell have the same sort of composition differences like, let's say, a brain cell? And if not, then it's not determined by the genes, it's determined by something else.
Right. Well, I think, Shahid, that is a fundamental question. I mean, what determines and regulates the composition of these biomembranes, right? I mean, first of all, the biomembranes are asymmetric. They have different lipids on the outer and the inner leaflet. And then they are different, their composition, meaning their relative weights of the different lipids, several thousand classes, several thousand different species are distinct in the different membrane compartments, meaning the cell surface is one compartment, the endosomes, the Golgi, the ER, mitochondria, other membranes. They all seem to be very distinct from each other. What maintains their distinction, and this is really remarkably tightly controlled. What maintains this distinction is one of the fundamental mysteries of cell biology. And it has to be addressed. I mean, we must begin to understand it. It is really, I think, something that is a wide open question. It is something that requires us to be able to understand how does a membrane sense its composition, right? How does that composition then translated into effecting some sort of a control on that process of sensing and an establishment? So it's a control theory question, and this is an incredibly exciting area where I think computer scientists, information processing people, and people who understand genes and their outcomes are involved. And the fact that, as you said, a liver cell and a brain cell have very distinct compositions, again, in all their internal membranes. One brain cell looks pretty much like another, or almost. And one liver cell, a hepatocyte, looks very much like a hepatocyte, another hepatocyte, but hepatocyte and brain cells look very different. And of course, different synthetic machineries are at play in both these cell types. And a combination of synthetic machineries, feedback, and control are likely to play roles. And in fact, this is something that we are very interested in pursuing, asking what are these controls and checkpoints that a cell has to ensure that it has a membrane of the right composition. I mean, if the membrane is the wrong composition, everything goes wrong. It doesn't matter what signals you send the cell, it's going to be read incorrectly. And we can also demonstrate that. So maintaining the right composition is crucial for life. Yeah. And it's a fundamental question, Shahid. Yeah.
Okay. Well, I don't see any other hands. So Jitu, it's late. We'll have to let you go. Oh, Shashi. Okay, Shashi will have the last word there. Go ahead, Shashi.
Okay, so I can now unmute myself. Jitu,
Yes.
more fundamental question, particularly based on the answer you gave to Shahid's question. Looks like the chemistry, of course, the chemistry determines the physics of the membrane with the physical properties. But it looks like it's the chemistry which sort of determines the function in such a way that its flexibility to accommodate very different proteins under different conditions, let's say temperature or ionic conditions. So it looks like the composition of membrane is so fundamental, and but there's still very few subtypes of the phospholipids, right? Compared to the membranes.
There are 5 subtypes of 5 phospholipid sort of classes, maybe 300 or 400 lipid types, which constitute these 5 classes. I mean, depending on the chain lengths, depending on the unsaturation, depending on the hydroxylation. There are over 300 to 500 to 500 different lipid species in the membrane of a given cell, and that's tightly related.
The lipid, the fatty acid chain length,
yes,
is extremely different in the neighboring domain. Will it sort of affect the physical properties, in terms of, let's say, very shorter version versus the very long version?
Absolutely. Because let's say you have a cell which has cholesterol. Cholesterol is pretty similar, across almost all eukarya. I mean, not exactly the same, but let's say all vertebrates. Now, if you have vertebrates that live at different temperatures, they have to measure their lipids against the length of the cholesterol molecule. And at cool temperatures and at warm temperatures, you need to have different chain lengths that can accommodate the membrane of the properties, the physical properties of a membrane, which are necessary for the functioning of various molecules in the membrane, for example, ion channels or even signaling systems. So I think the variation in the chain lengths and variation in the degree of unsaturation is something that is set by this medley between a measuring device and the possibility of synthetic complexity or synthetic control on these various lengths and degree of unsaturation. But it's reading some property that we don't understand. Okay, it is being read out and then controlled, right? But what is being read out and how is it controlled? I mean, how is it controlled may be easier to understand, but what is being read out, we just don't know right now. So, it's really fabulously exciting time. But given that we know that activity now is part of the equation, we know that, that the cell is actually going to regulate this, till kingdom come. Yeah.
Okay, Sarat first and then Krishna, and those will be the last two questions. Go ahead.
I think need to unmute, yeah.
Hello, I'm just asking this question in the context of B cells versus T cells, because B cells, let's say BCR interacting with this antigen. Let's say consider here 1:1 interaction versus T cells where we have PCRs and other receptors involved in this process. So how do you think this, this membrane clustering, which we have been actually, arguing that in T cells, once it encounters antigen, when a T cell becomes antigen experience, then TCR become clustered. But do you think the same thing would also happen in case of B cells? Because B cells, they are not really encountering many partners, they're very much, BCR versus antigen only.
Right. So let me, flip the question around. And I think both the B cell receptor and the T cell receptor would fall in the definition that I've just provided you into molecules that we would call active molecules, which are able to determine their own membrane environment. Now, T cell receptors, and of course, once you can determine what happens in your neighborhood and also determine and unleash, say, molecules like actin and actin polymerization machinery, as well as motor protein regulation, you can build, the proteins themselves can build the machinery that is optimal for their signaling outcome. And of course, that construction is determined by the outputs that these receptors have in the milieu of the membrane, right? So insofar as the B cell receptor and the T cell receptor engage with distinct machineries downstream in the cytoplasm, they're going to build different structures. And we already know that. There's some really nice work from Barbara Baird and David Holoka on the B cell receptor and their arrangement in the membrane upon engaging with the antigen. And there's actually a host of literature, including some work from the recently departed Katharina Gauss on the T cell receptor when it engages with antigen and the environment that the T cell receptor builds. So there's a lot to learn, and I think each receptor is going to reveal its own peculiarities and specialities, and there's perhaps going to be no general idea except the one that I mentioned, which is that these are active molecules and they will do their own thing. So they're building structures and using the membrane as an active player in their reading of information.
Krishna, go ahead.
Hi, Jitu. Thank you for an amazing talk. I had a small question about whether you expect to see or you see differences in kind of membrane dynamics, membrane environment, based on internal state, either of the cell, of the organism,
right?
That sort of thing.
Yeah. So I think this is where we are headed. We think we will see differences based on not only the state of the cell, but perhaps based on the cell cycle, or maybe the period, the clock.
Right.
Yeah. So I think because the synthetic machinery can also be regulated on these time scales, we're going to see differences and distinctions based on this, and that's going to be reflected perhaps in the composition of the membrane, and that will in turn influence the outcome of the cell.
Sure.
So and of course, any input that you give the cell in terms of diet, right?
Right.
That dramatically changes the outcome of receptors. So that we already know.
Right.
So you're right. I mean, the cell state, yes, but both the environment and the cell state together will contribute to what the membrane is actually reflecting, and that then contributes to outcomes. Yeah, signaling outcomes, I think.
Yeah, sure. Thank you.
Okay, Alok and Rabia, last two, absolute last two.
Hi, Jitu.
Hi, Alok. Hi.
Yeah. So this question is about worms. Many of the worms cannot make their fatty acids actually.
Oh.
So they have a very high level of fatty acid transporters, and they take in fatty acid from here. So I used to think that they probably have a very selected host range. But it turns out that they're not that selective in terms of what host there. So what do you think may be happening there?
Well, I mean, they might have enzymes that modify lipids, right?
That is possible.
Yeah. So once you can modify lipids, then you can regulate them, right? And regulate, I mean, maybe you can't make from glyceraldehyde 3-phosphate, maybe you can't make the entire thing, but if you got some way and then you can modify them, then those things can be under regulation. So I guess it would be important to ask what is the composition of these membranes under different diets, and what happens, I mean, under different sort of bacterial conditions, and see what happens. I mean, that would be really exciting. I mean, yeah. So this is a bit like invertebrates, and many insects actually don't make sterols, right? So they're sterol auxotrophs, and they require sterols for many things. So do worms. Some worms, I mean, C. elegans, for example. Yeah. So they require sterols, but maybe in a very different way than vertebrates.
Yeah, these are flukes mostly, blood fluke and others, liver flukes.
I see. But then they are swimming in a sea of lipids.
Yeah.
Yeah.
Okay, thanks.
Sure.
Rabia, go ahead.
Yeah, thank you, Professor Jamil. My question is an extension of what Professor Jamil actually put before some time back. Can we expect the similar kind of membrane dynamics to be happening in the cells of the nervous system, which do not allow too many disruptions to occur? But for the long-term memory processes, there are these membrane dynamics which are very important for the nervous system cells to function. So do we expect similar kind of nanoclustering in that also for the long-term memory processes? But otherwise, these nerve cells, they do not actually permit too many disruptions. But can we expect that similar kind of membrane dynamics, as you discussed, sir, in your lecture about the nanoclustering, that similar kind of nanoclustering is happening there also for the long-term memory processes of the brain?
Right. So I must say, I don't know much about how long-term memory processes are encoded in the nervous system, but wherever you find the actin, the myosin,
okay,
and relevant lipids, and many of these are actually downstream of some of the most famous metabolotropic receptors, some of the acetylcholine receptors. All these molecules activate the same set of actin players. And so I don't see any reason why, unless there is a way to shut it off, I don't see any reason why these sorts of membrane heterogeneities will not arise. And if they do arise, they will be used for something, I'm sure. So I think it's now a matter of looking and seeing what happens. But at the outset.
Yeah, I was just wondering that if this might be one of the ways that how brain cells are actually getting for long-term memory processes, because the short-term memory processes, they usually use the ion channels, but the long-term memory processes, they actually are into the cell signaling, deeper cell signaling processes that are actually involved there. So I was wondering that maybe nanoclustering is also something where which can be looked into as far as this function of the brain is concerned.
Well,
thank you, sir. Thank you.
I would certainly think it's a worthwhile thing to do, because we think this is involved everywhere. But of course, one has to be a little humble.
Thank you, sir.
Okay, well, thank you very much, Jitu, for this exciting talk, and thank you to the audience for joining us and for staying till the end. So thank you again, and look for the next announcement, as I said, on November 22nd. Thank you all. Thank you again. Good night.
Thank you, Shahid. Thanks, everyone.
Thank you. Bye.
Bye-bye.